Welcome to part 2 of sexual reproduction in flowering plants. In this lesson, we will explore the female reproductive structures and the process of pollination. We will understand the structure of the pistil and ovule, the formation of the female gametophyte, and how pollen grains are transferred to the stigma. Let us continue our journey into the fascinating process of reproduction in flowering plants. By the end of this lesson, you will be able to understand the structure of the pistil and ovule, explain the process of megasporogenesis and the development of the embryo sac, and learn about pollination and its different types. These concepts will help us understand how the female reproductive system of flowering plants prepares for fertilization. The pistil, also known as the gynoecium, is the female reproductive organ of a flower. It consists of three main parts: stigma, style, and ovary. The stigma receives pollen grains during pollination, the style connects the stigma to the ovary, and the ovary contains one or more ovules. Inside the ovule, the female gametophyte, known as the embryo sac, develops. Thus, the pistil plays an important role in the reproductive process of flowering plants. Now let us study the structure of the ovule, which is an important part of the female reproductive system in flowering plants. The ovule is a small structure present inside the ovary and is attached to the placenta by a stalk called the funicle. The point where the body of the ovule is attached to the funicle is known as the hilum. A typical angiosperm ovule is surrounded by one or two protective coverings called integuments. These integuments leave a small opening at the tip known as the micropyle. The micropyle serves as the passage through which the pollen tube enters the ovule during fertilization. The central region of the ovule is made up of a mass of cells called the nucellus, which provides nourishment to the developing embryo sac. Embedded within the nucellus is the embryo sac, which represents the female gametophyte of flowering plants. The region of the ovule opposite to the micropyle is called the chalaja. Thus, the ovule consists of important structures such as the funicle, hilum, integuments, micropyle, nucellus, chalaja, and embryo sac, all of which play vital roles in the process of sexual reproduction in flowering plants. Now let us understand the female gametophyte, also known as the embryo sac, which develops inside the ovule of flowering plants. The process begins with the megaspore mother cell, which undergoes meiosis to produce 4 haploid megaspores. Usually, 3 of these megaspores degenerate, and only one remains functional. The functional megaspore undergoes 3 successive mitotic divisions, producing 8 nuclei. These nuclei are organized to form the mature embryo sac, which is typically 7-celled and 8-nucleate, a characteristic feature of most angiosperms. At the micropylar end, there is an egg apparatus consisting of 1 egg cell and 2 synergids. The synergids possess special structures called filiform apparatus, which help in guiding the pollen tube into the embryo sac. At the opposite chalajal end, there are 3 antipodal cells. In the center of the embryo sac lies a large central cell containing 2 polar nuclei. Thus, a mature angiosperm embryo sac consists of 3 types of cells: the egg apparatus, antipodal cells, and the central cell with 2 polar nuclei. These structures play a vital role in the process of double fertilization and the formation of a new plant. Now let us understand pollination, an important pre-fertilization event in flowering plants. Pollination is defined as the transfer of pollen grains from the anther to the stigma of a flower. It is an essential step because it enables the male gametes present inside the pollen grain to reach the female reproductive structure. After reaching a compatible stigma, the pollen grain absorbs moisture and germinates to form a pollen tube. This pollen tube grows through the style and carries the male gametes towards the ovule, where fertilization takes place. Based on the source of pollen grains, pollination is of three types. Autogamy is the transfer of pollen grains from the anther to the stigma of the same flower. Geitonogamy occurs when pollen is transferred between different flowers of the same plant. Xenogamy is the transfer of pollen grains from the flower of one plant to the stigma of a flower of another plant of the same species. Among these, xenogamy introduces genetic variation, whereas autogamy and geitonogamy generally do not introduce new genetic combinations. Therefore, pollination acts as a crucial link between the formation of pollen grains and the process of fertilization, ultimately leading to the formation of seeds and fruits. After pollination, the pollen grain reaches a compatible stigma and begins the process of germination. The pollen grain absorbs moisture and nutrients from the stigma, causing it to become active. The intine, which is the inner wall of the pollen grain, protrudes through one of the germ pores of the exine and forms a pollen tube. This pollen tube grows through the tissues of the style and carries the male gametes towards the ovule. The growth of the pollen tube is guided by chemical signals released by the synergid cells present in the embryo sac. The pollen tube usually enters the ovule through the micropyle, a process known as porogamy. Once the pollen tube reaches the embryo sac, it releases the two male gametes. One male gamete will fuse with the egg cell to form the zygote, while the other will fuse with the two polar nuclei to form the primary endosperm nucleus. This process, called double fertilization, will be studied in detail in the next part of the chapter. Thus, pollen germination is an important step that connects pollination with fertilization and ensures the successful transfer of male gametes to the female gametophyte. Double fertilization, which is a unique and characteristic feature of flowering plants or angiosperms. After the pollen tube enters the ovule through the micropyle, it reaches one of the synergid cells and releases two male gametes into the embryo sac. The first male gamete fuses with the egg cell to form a diploid zygote. This process is known as syngamy. The zygote later develops into the embryo. The second male gamete fuses with the two polar nuclei present in the central cell to form a triploid primary endosperm nucleus, also called pen. This process is known as triple fusion. The primary endosperm nucleus develops into the endosperm, which provides nourishment to the developing embryo. Because two fusion events occur in the same embryo sac, syngamy and triple fusion, the overall process is called double fertilization. Double fertilization is a remarkable evolutionary adaptation of angiosperms because it ensures the simultaneous formation of both the embryo and the nutritive endosperm, which are essential for successful seed development. After double fertilization, two important structures begin to develop inside the ovule, the endosperm and the embryo. The primary endosperm nucleus, pen, which is triploid in nature, undergoes repeated mitotic divisions and forms the endosperm. In most angiosperms, the early development of endosperm is free nuclear, where nuclear divisions occur without immediate formation of cell walls. Later, cell walls may be formed, resulting in a cellular endosperm. The endosperm serves as a nutritive tissue and provides nourishment to the developing embryo. At the same time, the zygote formed by syngamy develops into the embryo. The zygote undergoes repeated mitotic divisions and passes through different developmental stages such as the proembryo, globular stage, and mature embryo. A typical dicot embryo consists of an embryonal axis and two cotyledones. The upper end of the embryonal axis is called the plumule, which develops into the shoot system, while the lower end is called the radicle, which develops into the root system. In monocote plants, the embryo possesses a single cotyledon known as the scutellum. Thus, after fertilization, the ovule develops into a seed, and the ovary develops into a fruit. The seed contains the embryo and stored food, ensuring the continuation of the plant life cycle. After fertilization, remarkable changes take place in the flower, leading to the formation of a seed and fruit. The ovule develops into a seed, while the ovary develops into a fruit. The wall of the ovary transforms into the fruit wall, known as the pericarp. The pericarp may be fleshy, as seen in fruits like mango and tomato, or dry, as seen in pea and gram. Inside the developing seed, the zygote develops into an embryo, and the primary endosperm nucleus gives rise to the endosperm, which provides nourishment to the developing embryo. The protective coverings of the ovule, called the integuments, develop into the seed coat, which protects the embryo. Depending on the presence or absence of endosperm in the mature seed, seeds can be classified as endospermic seeds and non-endospermic seeds. Endospermic seeds retain a part of the endosperm in the mature stage and provide stored food for the embryo. Examples include maj and castor. In non-endospermic seeds, the endosperm is completely consumed during embryo development. Examples include pea and bean. Thus, the formation of seeds and fruits represents the final stage of sexual reproduction in flowering plants. Seeds ensure the continuation of the species by giving rise to new plants under favorable conditions. Let's quickly revise the important concepts from this chapter. The female reproductive structure in flowering plants is called the pistil, which consists of the stigma, style, and ovary. Inside the ovary are ovules, and each ovule contains an embryo sac. The mature female gametophyte, or embryo sac, is 7-celled and 8-nucleate, making it highly specialized for reproduction. Pollination is the transfer of pollen grains from the anther to the stigma. After pollination, fertilization takes place. Angiosperms show a unique phenomenon called double fertilization, where one male gamete fuses with the egg cell and the other fuses with the polar nuclei. Following fertilization, important post-fertilization changes occur. The ovule develops into a seed, while the ovary develops into a fruit. Thus, sexual reproduction ensures continuity of life and creates genetic variation in flowering plants. With this, we complete the major concepts of Part 2. Congratulations on successfully completing Part 2 of NCERT Class 12 Biology, Chapter 1. In this session, we explored female reproductive structures, the ovule and embryo sac, pollination and pollen germination, double fertilization, and the development of seeds and fruits. These concepts form the foundation for understanding reproduction in flowering plants and their life cycles. Remember, knowledge grows when curiosity blooms. Keep asking questions, keep exploring, and keep learning. In the next session, Part 3, we will study post-fertilization events and apomixis in detail. If you found this lesson helpful, don't forget to like, share, and subscribe to Rush Learning Hub. Thank you for learning with us. Until next time, keep rising, upgrading, shining, and honoring your potential.